Alright, hi everybody. My name is Brandon Ng. I am a junior this year, and I'm here today to talk about my project, which is other pathogenic vibrios in the ROI Canal. And I'm here to just talk about what I did over the year. So first off, since we're talking about the ROI Canal, we have to go over some history of it. So first of all, the ROI Canal was built in 1928, and its intended purpose was to create more land near downtown Honolulu for a new community, which is we know it now as Ala Moana and Makiki area, and it drained a lot of the swamplands in Makiki. As you see in the picture on the left, that is a picture of the Moana Surf Rider in 1920. So this is before the ROI Canal was built. And as you see on the top of the picture, that there's like farmlands and wetlands, that would be drained later on, because on the right it is a fast forward in time to 1927, when the Alawai Canal is built and almost ready to and almost completed. That that farmland you see on the left picture, even though it's not exactly like perfect picture as the picture's been shifted to the left, you can see that that farmland is no longer there. It's dry. It's able to be built on, and today it is full of waste and harmful bacteria. And some of the harmful bacteria that I'm talking about today is Vibrios, more specifically Vibrio cholera and Vibrio perihemolyticus. However, not all Vibrios or perihemolyticus are pathogenic or cause disease. In the case of Vibrio cholera, it only causes the disease that we know as cholera due to two serotypes called O1 and O139, and Vibrio perihemolyticus only causes disease due to one serotype called O3K6. A serotype is a distinct variation within a species of bacteria due to cell surface antigens. And these Vibrios, they like living in brackish and warm water. In the case of Vibrio cholera, it is known as a freshwater disease, and it causes symptoms like watery diarrhea, dehydration, nausea, and vomiting. However, even though it is known as a freshwater disease, research has shown in a paper from 1982 that Vibrio cholera actually thrive in higher salinities, like and with higher temperatures, like we have here in a temperate region. And for Vibrio perihemolyticus, that causes bloody diarrhea and vomiting, they thrive in environments also with higher temperatures, but instead with a low salinity. And Vibrios cause clinical forms of the disease by itself, though, as I mentioned before, these bacteria don't cause the disease, but some serotypes of the bacteria can cause the disease. In the case of Vibrio cholera, the bacteria must have a toxin, which is from a filamentous phage called CTX5, that also contains the CTX-AB operon. That CTX-AB operon encodes for the A and B subunits of the cholera toxin. That toxin causes the disease that we know as cholera. And in Vibrio perihemolyticus, the bacteria must have the two hemolysins. A hemolysin is a lipid or protein that can cause the lysis of red blood cells, and the two hemolysins are called thermostable direct hemolysin and TDH-related hemolysin. The hemolysin TDH and the TDH-related hemolysin are only found in clinical samples like Vibrio perihemolyticus O3K6. And the picture on the right is a picture of the Vibrio cholera, how it would get how it gets its like pathogenicity. As you see that on the top of the picture where it says CTX5, that's part of the phage, and in the phage, when it actually goes into the bacteria, it'll actually like reverse reverse RNA, take part of its DNA and encode it into the bacterial DNA, and that's how you get the operon in the bacteria. Though you might be saying, well, this all sounds horrible, but why do we care about this in the first place? Well, first of all, the RA Canal is in the heart of Waikiki, and people use the canal for recreational activities like canoeing and paddling, and also for other public safety reasons, like helping the government inform the public when the ROI is the most dangerous. Because right now, especially for like Iolani students that are in like paddling and canoeing, you might be falling into the ROI when the ROI is the most dangerous and you will never know it. So I think this type of research is very important for public safety health. And with all that research, my research question is, what is the abundance of pathogenic Vibrio cholera and Vibrio perihemolyticus, and how much of a threat are these bacteria to people that use Yalawai? So first of all, you have to get the samples. So I got the samples by first making a map of the Yalawai and then going down and sampling water with bottles. Next, I brought the bottles back into the lab and I filtered the water by making 1:1, 1:10, and 1:100 dilutions. And then I poured the dilution into 0.45 filters, and then I took those filters and plated it on TCBS and chrome agar. TCBS and chrome agar are two agar mediums that are used to specifically grow Vibrios. So an example of a TCBS plate would be on the left side, which is a green. It's supposed to be green, but this is with sample, so it appears kind of yellow in the picture. And on the right of that, it is chrome agar Vibrio, and it's an orange yellowish plate when you first make it. And on TCBS, the yellow colonies are Vibrio cholera, and Perihemolyticus and Vonificus appear green. And on chrome agar, mauve is Perihemolyticus only, and Vonificus and Vibrio cholera are blue. So after I plated the filters, I had to let the samples incubate for 24 hours to let the bacteria grow, let the colonies grow. And then after, I came back the next day and we picked the colonies that were supposed to be Vibrio cholera and Vibrio perihemolyticus. On TCBS, I only picked the yellow colonies because, as I mentioned before, the yellow colonies should identify as Vibrio cholera. And on chrome agar, I picked the mauve colonies because mauve should be Vibrio perihemolyticus on chrome agar. So after I had all of these colonies and I picked the colonies, I had to extract the DNA from it. So the picture to the and then after, we use different primers to identify different things that we were looking for. So because every bacteria has different primers that you use in a PCR, and each bacteria has different genes. So for Vibrio perihemolyticus, we first had to use TLH primers, or thermolabile hemolysin, which is found in all perihemolyticus, and it's used to identify just if you have perihemolyticus in general. Then we would run a gel electrophoresis, as you see in the picture on the bottom. So when you see, so for gel electrophoresis, right, these are what you would call the negative samples, or as we call primer dimer, because primers in a PCR, which is explained in the prime recognition, if there is if there's no DNA there and the primer does not recognize that there is DNA, it would actually just rather amplify over itself. And there's not a lot of, and gel electrophoresis is run to positive to negative, and that the base pairs that are the least, they run the farthest. So when the primers overlap over each other, they run pretty far because there is no base pairs to hold down speed. And on the case of samples 1, 9, 10, and 11, which is, there actually is bacterial amplification there. So then, how many samples were actually pathogenic? Well, we found that TLH, when we ran the TLH on PCR and gel electrophoresis, we found that on site 1, site 2, and site 3, there was actually a pretty decent amount, though it was kind of low, about 20% on average of identifying Vibrio perihemolyticus, and we didn't find any actual pathogenic Vibrio perihemolyticus. But for cytotoxic, or CTX, which is for cholera, we found that on site 1, which is the corner of the Manoa stream near Iolani School, we found there was very few, and also near the Alawai Park, that's near Alawai School. But for the storm drain, we found that there was actually a pretty significant amount, as 30% of the samples were actually cytotoxic. So all in all, we found no pathogenic Vibrio perihemolyticus in the Alawai Canal. However, we did find that there was a presence of it. So even though we did find a presence, I can say that there is no actual threat to human health, because environmental or non-clinical bacteria don't cause any threat to humans. However, Vibrio cholerae is a threat in the ROI, but it's mostly centered around the storm drains. And a reflection on what I learned this year is that during my research, I learned that being organized is a big part of being a good researcher. It helps if you keep a folder or a binder to keep your lab work, because it makes it easier, especially when you're trying to compile your data to make a graph. It's hard if you have your papers in like 3 different places and they're not ordered correctly. It makes it easier for you to compile your data, and especially if you need to make changes, like when I was running my PCR, sometimes the primers don't work as well. If you don't keep it organized, there's no way you would be able to identify that and make changes to make sure that your project would go smoothly. So I'd like to say thank you to Dr. Chan for giving me a chance to do research in the lab, and also for being my mentor and helping me out. And Mr. Eric Tong for helping me with the DNA extraction. And my mom, dad, and brother, my mom and dad over there, for helping to drive me every day and for putting up with me complaining about how research is not going as well as I thought it would. And thank you for spending your time today to listen to my speech. I'm sorry.